Intermittent fasting has emerged as one of the most promising non-pharmacological strategies to activate autophagy, a cellular renewal process that removes damaged organelles, misfolded proteins, and accumulated waste. By engaging in daily or periodic fasting periods, most commonly between 16 to 24 hours, the body enters a state of nutrient deprivation that downregulates insulin signaling and mTOR activity while upregulating EMPK. This biochemical shift tells cells that it's time to conserve resources and clean house. Autophagy, which literally means self-eating, is then activated as a natural cellular recycling program. The benefits of this process are vast and include enhanced metabolic efficiency, reduced oxidative stress, and potentially slower aging at the cellular level. Nutritionally, it's best to consume only water and electrolytes during fasting periods to maintain hydration and support nerve and muscle function without interrupting autophagy. Interestingly, the body becomes more metabolically flexible over time, which may enhance both fat oxidation and insulin sensitivity. Unlike calorie restriction, intermittent fasting offers a sustainable and cyclical approach, allowing cells to oscillate between stress and recovery, much like muscles grow stronger after rest post-exercise. Exercise is another powerful trigger for autophagy, particularly when done in a fasted state. During moderate to intense aerobic activity, cells experience a temporary energy deficit, which activates AMPK and inhibits mTOR in a similar manner to fasting. This induces a wave of autophagic activity that helps muscles remove damaged mitochondria and other cellular debris, leading to improved endurance and muscular function over time. The muscle fibers essentially clean and rebuild themselves for greater efficiency. Regular physical training doesn't just stimulate autophagy in muscle, though, it also enhances brain autophagy, which may explain part of the cognitive benefits of exercise. To support this, amino acids like leucine from whole foods rather than supplements can help rebuild tissue post-exercise without bluntly suppressing autophagy if timed appropriately, ideally after the fasting window or workout ends. Polyphenol are another category of compounds that have been repeatedly associated with enhanced autophagic activity. Found abundantly in plant-based foods, polyphenol like resveratrol from red grapes, quercetin from apples and onions, and EGSG from green tea modulate key signaling pathways such as SIRT1 and AMPK. These compounds are thought to mimic the effects of caloric restriction, acting as mild cellular stressors that nudge the body toward autophagic renewal. What's compelling is that polyphenol don't need to be consumed in massive doses to be effective. Consistent, modest intake through a colorful, plant-rich diet seems to be enough to promote cellular health. Green tea, in particular, provides a double benefit due to its mild caffeine content, which also enhances fat oxidation, and its antioxidant properties that reduce inflammation while permitting low-level stress responses essential for hormesis. Another nutrient that has garnered interest is spermidine, a naturally occurring polyamine found in aged cheeses, soybeans, mushrooms, and fermented foods. Spermidine has been shown to induce autophagy independently of fasting or caloric restriction by influencing acetyltransferase activity and modulating autophagy-related genes. In studies on both animals and humans, increased spermidine intake has correlated with improved cardiovascular function, reduced inflammation, and even increased lifespan in some models. This makes it a particularly exciting nutritional candidate for long-term autophagy support. Importantly, its mechanisms appear to be distinct from those of AMPK or mTOR, suggesting that it could synergize with fasting or exercise-induced autophagy in a complementary way. Sleep also plays a significant role in regulating autophagy, particularly in the brain. During deep, slow-wave sleep, the glymphatic system becomes more active, clearing out metabolic waste and supporting neuronal health. This cleansing action appears to be tied to autophagic processes in neural tissue. Poor sleep, on the other hand, impairs autophagic flux and is associated with the accumulation of toxic proteins like beta-amyloid. Therefore, establishing strong circadian rhythms and sleep hygiene not only supports cognitive performance, but also enhances the body's ability to maintain cellular homeostasis. Magnesium, found in leafy greens, legumes, and seeds, is a mineral that supports sleep quality and also plays a role in cellular energy regulation, thereby supporting autophagic pathways. Finally, mild thermal stress, such as exposure to cold or heat, may activate autophagy through different stress response mechanisms. For instance, sauna bathing increases heat shock proteins and promotes autophagic repair in various tissues, while cold exposure can stimulate brown fat activity and metabolic changes that trigger EMPK. While still a developing field, these forms of hormetic stress appear to share common cellular signals with fasting and exercise. Nutritionally, adaptogens like rhodiola or reishi mushroom might support the body's resilience to thermal stress while also providing antioxidant and autophagy modulating effects. In essence, autophagy is not activated by deprivation alone but by rhythm and intelligent stress. When fasting, exercise, phytonutrient-rich diets, proper sleep, and environmental challenges are woven together thoughtfully, the result is a dynamic internal environment where old cells make way for the new. Rather than merely preventing disease, this approach to cellular renewal may lay the foundation for true health span, years lived not just longer, but better.